In this section of our notes, we're going to focus on the plasma membrane and the extracellular matrix. And first we're going to start with the plasma membrane and go through some general functions. So that plasma membrane is what separates the internal from the external environment. It's the outermost part of the cell. It helps regulate what moves in and out of the cell. It's a semi-permeable, or we could also say selectively permeable membrane. Some substances can cross it and others cannot. It helps the cell maintain homeostasis, and that is a constant internal environment. And as a result, it also helps maintain the homeostasis of the entire organism, and it allows for communication. Cells can communicate with each other, and cells can also communicate with the environment. And then it also adds in the adhering of cells, or the anchoring. Most of your cells aren't floating around. They're anchored together, forming tissues. Now, we've already mentioned that there's many molecules that come together to form the cell membrane. And these molecules interact due to the nonpolar and the polar regions that they possess. Now, the model of the cell membrane is called the fluid mosaic model. Now, in this model, you're going to see that molecules, or parts of molecules that are polar, are going to be on the exterior sides of the membrane. So what I mean by exterior sides is just not on the outside of the cell, on the exterior part of the membrane. So all of these molecules that you see here are going to be polar, or these regions are polar. If you remember, cells are surrounded by an aqueous environment, which means it's mostly water. And water is a polar molecule as well. And polar molecules want to interact with other. When you look at the side of the membrane that is bordering the cytoplasm, then all of these molecules that we see here are going to be polar, or those regions of those molecules are going to be polar as well. Again, cytoplasm is an aqueous environment. It contains mostly water. So those polar regions of these molecules are going to be interacting with the water in the cytoplasm. So that is what we mean by the exterior side. So here's an exterior side, and here's an exterior side of the membrane. The molecules on the internal part of the membrane, so right here, are hydrophobic, or nonpolar. These nonpolar molecules interact with themselves and do not interact with polar molecules. Now we're going to go through the different molecules that make up the plasma membrane. And some of them we've already discussed before. So let's start with the phospholipids. Now, we've already said that there's two parts to the phospholipids. They have the nonpolar tails that you see here, and then they have the polar head. So remember, that head is hydrophilic, which means water-loving, and those tails are hydrophobic. Because of this property, two layers of phospholipids form to form that membrane. So we say that they're arranged in a bilayer. Now, something that is new, because we didn't discuss this in pre-AP, is that phospholipids aren't stationary. They can actually move. So these phospholipids can change places, and these can change places, but a phospholipid can't flip from one side to the other. So you might put an X and indicate that they can't flip. Now, we've talked about cholesterol before. It is a lipid steroid, but unlike other fats, it has both a nonpolar and a polar region. So that explains why it's located, where it's located in this membrane. So here is a cholesterol molecule, and you can see that they've denoted two different parts. So they're indicating this part is a little bit different from this part. So that top part, which is close to the heads of those phospholipids, is going to be polar, and it interacts with other polar molecules. Whereas the carbon rings of this steroid are nonpolar. So they're embedded into the internal part of the membrane, and they're interacting with other nonpolar molecules. We discussed that these cholesterol molecules make that membrane more fluid, in colder temperatures, but less fluid in warmer temperatures. The next molecule, the glycoproteins, I've mentioned briefly. They are proteins with a carbohydrate chain attached. And there's quite a few in this diagram. So I can tell that there's a protein part right here, and then you see that there's this chain-like structure that's hooked to it. So there is a glycoprotein. So you see that it's made of one part protein and one part that carbohydrate chain. Here's another glycoprotein. Now, there's many different types of glycoproteins with many different functions. So in general, these glycoproteins that are found on the membrane serve as markers to help identify the cell type. Some of them are receptors. Molecules like hormones can bind to these glycoproteins. And since those chains, those carbohydrate parts, are polar, so let's kind of indicate that, they're interacting with water and they're helping to stabilize the structure of the membrane. Glycolipids are along the same line as these glycoproteins, except you have a lipid with a carbohydrate chain attached. And I can find a glycolipid right here. So you can see that there's a carbohydrate chain and it's attached to the head of that phospholipid. Again, there's lots of different types of glycolipids, but in general, the ones that are embedded in the membrane can serve as receptors or markers for the cell. Next, we're going to focus on proteins, and most of the lecture is about proteins, the different types of proteins, and the different jobs that they perform for the cell. Now, just like phospholipids have nonpolar and polar regions, we learned that proteins have polar and nonpolar regions as well. And the nonpolar regions fold more on the inside of the protein, and the polar regions fold more outward to interact with water. And these nonpolar and polar regions affect how the proteins are inserted in the membrane, as you can see here. So you can tell that this part of the protein is going to be nonpolar because it's interacting with the nonpolar tails. Whereas these parts of the protein that are more on the external part of the membrane are going to be polar regions. Now I'm going to move back up a little bit so that I can reference the diagram at the top so you can understand what I mean by integral proteins and peripheral proteins and transmembrane proteins. Let's relate this idea of peripheral proteins to peripheral vision. You've probably heard of that. You can use your peripheral vision to see things that are not directly in front of you. So peripheral means on the outside. So proteins, like this one right here, that are on the exterior of the cell membrane are called peripheral proteins. And hopefully you've caught on to the idea that since they're on the external part of the membrane, then those are going to be mostly polar proteins. Proteins that are embedded into the phospholipid bilayer are called integral proteins. And you can see that here. This protein is integrated into the phospholipids. And then integral proteins that go all the way across the membrane from one side to the other are called transmembrane. Trans means to spread or go across, so transmembrane. So this integral protein that you see right here is also a transmembrane protein because it's going all the way across the membrane. Now, before we get into the specific jobs of different types of proteins in the membrane, I just want to highlight this diagram because I think it's a great diagram to help you compare some of the different jobs that proteins of the membrane have. So we have transport proteins, and you can tell that transport proteins are moving molecules across the membrane. These proteins are involved in joining cells. Here we have an enzymatic protein. So what this is showing you is that we're starting off with this molecule, molecule A, and this enzyme is converting it into molecule B. And then the second enzyme is taking molecule B and converting it into molecule C. So many enzymes that assist in chemical reactions are embedded in a phospholipid bilayer. And then we have receptor proteins in the membrane that can bind to signaling molecules. And the term for signaling molecules is called ligands. So we're seeing some kind of molecule like a hormone that's binding to this receptor protein here, and that's going to send a signal for the cell to respond in some way. Here's a situation where there's a protein and it is being utilized to recognize this cell, as in it's a cell that belongs to you. This is most likely a white blood cell that's binding to one of your cells and just making sure that it's one of your cells and it's not a foreign cell. And this is what I mean by there's proteins that aid in anchoring, or the cell membrane helps to anchor cells. So we have a protein right here, and it's bound to cytoskeleton on the inside, and it's bound to proteins and carbohydrates and other molecules on the outside of the cell, and that's helping to anchor the cell in place. Now, the table that I created here is trying to summarize some of the information that you need to know about these different types of proteins. And I divided it into information that you already were taught in pre-AP and some additional information that you need to pick up from this lecture. But I don't want to talk about all this information right here because I like to refer to diagrams and charts to help you better understand some of these concepts. So let's move to the first type of protein, the transport proteins. And as you can gather from the name and from these diagrams, that these are proteins that transport molecules across the membrane, either from the outside of the cell to the inside of the cell, into the cytoplasm, or from the cytoplasm to the outer part of the cell. We say that these proteins are molecule specific. And what that means is that proteins can usually only transport 1, 2, 3 different types of molecules, and that's it. And only certain types of substances can move across a certain type of protein because of the shape of the protein and the shape and the properties of these molecules that are trying to cross. An example is with aquaporins. Here's a picture of an aquaporin. An aquaporin allows water to cross the membrane, but it also allows a few other molecules to cross. But nonpolar molecules would not be able to cross through an aquaporin due to the interaction of the molecules of the aquaporin and of these nonpolar molecules. Now, there's 3 main types of transport proteins, so sometimes instead of saying transport protein, I'll say a carrier protein or a channel protein or a pump. And here's the reason why these proteins are referred to by different names. If we have a channel protein like this one right here, it's always open. So you can see that this molecule is always open, allowing, in this case, protons or hydrogen ions to cross or flow. In this situation, molecules are going to be flowing from high concentrations to low concentrations passively. So they move from high concentrations to low concentrations. Now, carriers, which you can see right here, are proteins that move molecules across the membrane by changing shape. So what happens is whenever a molecule, in this case a proton, binds to this carrier protein, that binding causes a shape change. And that shape change results in the opening into the cytoplasm in this case, and that ion crossing the membrane. In this situation as well, molecules are moving from high concentrations to low concentrations. So there's a low concentration of protons inside of that cell. When we're referencing a pump is moving molecules against the gradient. Moving molecules from low concentrations to where they're in high concentrations. So you can see that there's a higher concentration of protons here compared to the inside. But this pump, this transport protein, is moving that proton from low concentrations to high concentrations. Now, these next concepts aren't super important, but I don't want a diagram like this to throw you if you see something like this on a test. I want to point out that sometimes proteins are moving 2 molecules at a time, and that's what you're seeing right here. When they're just moving 1 molecule at a time, it's called a uniport. In this situation, 2 molecules are being moved at once, and in this situation, they're going in the same direction, but they can also be moved in opposite directions. So when this molecule is moved across, then this transport protein will bind to the other type of molecule and move it in the other direction. So I just wanted you to be aware that this can occur with transport proteins in the membrane as well. And then finally, let's mention this really important transport protein, the aquaporins. Aqua means water. So again, here's the aquaporin. It's a protein that's embedded in the membrane. It's a transmembrane protein. And this protein allows water to cross. Because remember, water is a polar molecule, and it cannot cross through the tails, which are nonpolar molecules. And so one reason why water is able to cross through this aquaporin is because when this protein folds up, it creates this positively charged center. And remember that there's charges associated with these polar water molecules. And so those negative oxygens are going to be attracted to this polar center. So these molecules, the water and the aquaporin, will be able to interact, and those molecules will be able to cross through the membrane. Next, we have marker proteins. And we said that many marker proteins are glycoproteins. So there's a protein component and a carbohydrate chain. And these proteins help cells identify self cells, as in cells that belong to you, and non-self cells, foreign cells like bacteria cells. Now this diagram is showing you that here is the marker protein. There's a marker protein on one of your body cells, and your body cells recognizes that marker protein. But if you have a bacteria cell, it has markers as well, but those markers are not going to be recognized by the body. As a result, the body's going to try to destroy those non-self cells. And that's what this diagram is attempting to show you, that when a cell is recognized as being foreign, then your body has an immune response. And in this case, your body has produced antibodies to fight the infection or to kill the pathogen. But you can see that these antibodies, which are these Y-like structures, have only bound to the bacteria cell, and they're not binding to your own cells that make up your body tissues. But sometimes your body doesn't recognize its own cells. Here's a situation where the body has created antibodies, and those antibodies are even targeting self cells. When your body doesn't recognize certain cells as self cells and destroys them, then you have what's called an autoimmune disorder. An example of an autoimmune disorder is type 1 diabetes. For unknown reasons, what happens in this instance is that the immune system attacks and kills these cells that produce insulin, these beta cells that are found in the pancreas. And as a result, this person can no longer produce insulin for the rest of their lives, and they have type 1 diabetes. This person can be totally healthy one day, and then the next day, as a result of your immune system waging battle on your own cells, then you can develop this type of disorder like type 1 diabetes. And now we're going to talk about receptor proteins. Most of what we've talked about so far in regards to receptor proteins are the concepts related to this first diagram, that there's chemical messengers, or there's ligands that can bind to these receptor proteins and send a signal to the cell. But I want to point out that there's many different substances and other cells even that can bind to receptor proteins. That's what this diagram is attempting to show you, that molecules that are considered toxins can bind to receptor proteins. Viruses gain access to your cells by binding to these receptor proteins. Bacteria can bind to receptor proteins. And then even other cells in your body can bind to receptor proteins of a different cell. And remember, we said that many marker proteins are considered glycoproteins. But remember, we also have those glycolipids, and that is a lipid with this carbohydrate chain attached. And they can also serve as cell receptors. But the common idea to understand is that whenever a substance binds to a receptor, then the cell is going to respond in some way. Down below, I've included some diagrams that attempt to show you some of the responses that can result due to the binding to these receptors. In the biochemistry unit, we talked about ATP and the fact that 2 phosphates can be removed from ATP, and it results in a molecule called cyclic AMP. And cyclic AMP is known as a secondary messenger. And the secondary messenger spreads the signal to other molecules that are in the cell. So whenever this ligand, or this signaling molecule like a hormone that you see here, binds to this receptor, then it activates the making or the synthesis of the cyclic AMP molecule. You can see once activated, these cyclic AMP molecules are going to cause the cell to respond in some way. In this diagram, I'm attempting to show you how viruses gain access to your cells using receptor proteins. So here we have a virus, and it's binding to a receptor. And the response in this case is the cell will take in this virus. So the genetic information, either RNA or DNA that's found in the virus, let's put DNA or RNA, has now been injected into this cell. And that will ultimately lead to the production of new viruses by your cell that will leave the cell and will likely cause cell death. In this last diagram, I'm showing you how a cell responds whenever it binds to a bacteria cell. So in this situation, we have a B cell, and a B cell is an immune cell. And we have an entire unit dedicated to immune cells, like B cells and T cells. But we have a receptor protein on the surface of our immune cells, like our B cells, and they can bind to bacteria. And when a B cell binds to a bacteria, this is the response. In this situation, the cell starts to divide or proliferate, form more B cells basically. But B cells can be signaled to divide whenever chemicals bind to them. So here's a molecule, it's called a cytokine, that's been released from another white blood cell, another immune cell. And when this chemical binds to receptor protein, so I'm going to draw a receptor protein on that B cell, then that too can also cause this B cell to divide and proliferate and form an army of B cells that can help fight this infection. Now, so far I've just indicated that there's receptor proteins and cells, bacteria, viruses, molecules can bind to those receptor proteins and cause a response in the cell, but of course there's way more to it. Now, don't go digging through your notes for this page, but this is a page that is in your biochemistry note packet where we talked about the 4 big ideas. And I want to point out big idea 3, that living systems store, retrieve, transmit, and respond to information. So we've talked a lot about retrieval, as in receptor proteins retrieve information whenever some kind of signaling molecule binds to them. But I want to talk more in depth about the transmitting and the responding part. And I also mentioned that when you think of big idea 3, I want you to think of communication. So when we're talking about the retrieving and transmitting and responding to information, we're talking about how cells can communicate with one another. How information from other cells or how information from another part of the body can be received by cells in a different part of the body, causing a response there. Now, the retrieving of information, the transmitting of it to other molecules in the cell, and then finally the response by this cell is what we mean by a signaling pathway. So first with reception. As we mentioned, there's going to be a receptor protein that can bind to some kind of signaling molecule. And remember, the general term for a signaling molecule is called a ligand. And initially, whenever this ligand binds to this receptor protein, what's going to happen is the receptor is going to have a shape change. And that's a big concept to understand, that this shape change is going to allow for other molecules inside of the cell to become altered. In this diagram, it's the shape change that is going to allow this protein that is inactive to become active. So think of it like this. There was a signal that was brought to the outside of the cell, and now that signal has been relayed to the inside of the cell, to these molecules that are in the cytoplasm. Now, this newly active protein is going to start off a series of reactions. And this newly active protein is what is often referred to as a secondary messenger. Like cyclic AMP. So we had a primary messenger, which could be a toxin, could be a hormone, could be an ion, that is ultimately leading to the activation of this secondary messenger. From here, a signal seems to be spread out through the cell, from molecule to molecule. And that is what we mean by transduction, that the signal is being transducted from one molecule to the next. And eventually this signal is going to reach some target in the cell, and it just depends on which hormone or signaling molecule activated which receptor protein. But targets include things like enzymes. They can become activated, or organelles can be affected, or transport proteins can be altered, or transcription factors. In this diagram here, the target is a transcription factor, which is found right here. Now, remember, in the process of transcription, an mRNA molecule copies the gene on DNA, and as a result, eventually, we're going to get a protein produced. So this leads to protein production. And a transcription factor is any molecule that can affect this transcription process. So before this was active, whenever this transcription molecule was inactive, transcription wasn't taking place. But whenever it becomes active, then transcription is taking place, and the gene is being expressed in this situation. Now, during transduction, or the spread of this signal throughout the cell, amplification is occurring. And amplification is the growth of this signal. And here's what I mean by the signal becomes amplified. In this diagram here, you can see that this molecule is activated, and it is activating all of these cyclic AMPs. I believe there's 7 of them there. And each cyclic AMP can activate many molecules itself. I think it shows that the cyclic AMP is activating 7 different protein kinases, and each one has the ability to do this. So the signal is growing because the number of molecules that are activated is increasing exponentially, because one molecule can activate many. And then eventually the cell is going to respond in some way. So there's the response part of this signal transduction pathway. And I already mentioned that targets like enzymes can become active, or an organelle can release stored ions which would affect another part of the cell, or transcriptions become active and a gene is expressed, or membrane gates open and ions go moving out of the cell or come rushing into the cell. So there's many different type of responses, let's change that can occur simply due to a signaling molecule or some kind of signal being received by a receptor protein that's on the membrane of the cell. Now, I can tell you from analyzing past tests that cell signaling is a big deal. You will find a large portion of your multiple choice questions are going to be about some kind of signal transduction pathway, or you're going to have short answer questions or your essay in which you're going to have to explain what occurs during a signal transduction pathway. So make sure that you can tell me there's 3 stages of the signaling pathway. There's a reception, there's transduction, and there's a response. And you should be able to describe, in general, some of the processes or activities that take place during each of these 3 stages. You don't have this page that I'm showing you now, but I just want to point out what you're supposed to know about these different receptor proteins and some of the signaling transduction pathways that can occur in cells. This is the course description guide, and this tells me what I'm supposed to teach you guys. That a receptor protein recognizes signal molecules causing the receptor's protein shape to change, which initiates transduction of the signal. And I can pick one of these to help facilitate or teach that concept to you. And I'm about ready to teach you this idea with these G protein linked receptors. Now, even if I don't teach these other ones to you, no guarantee that you won't have a question about them. If you see a question about ligand-gated ion channels like you see here and the receptor tyrosine kinases, they're going to give you a lot of background, but you're going to have to read that information, basically teach those concepts to yourself right then and there during the test and answer questions about them. So I'm going to teach you about all 3 of these, but I'm going to break it up, because whenever there's similar concepts, students have the tendency to mix all the different parts between or among these different concepts. So I'm going to talk about these whenever we get into the nervous system. We're talking about nerves. And then I'm going to talk about these receptors when we're talking about cell division. So my goal is just to teach you one of these ideas at a time so that you'll better understand it. So that's what we're going to tackle now. So I wanted to explain to you why I'm getting so in depth here. It's because this concept can be really confusing to students if they're just reading about it for the first time and having to answer questions about it. So we're going to look at a signal transduction pathway that utilizes a G protein linked receptor that's bound to the membrane of a cell. And we're going to talk about a very specific type of cell in the body, a liver cell. If you remember in the biochemistry unit that whenever animals consume excess glucose, they store it as glycogen in their liver cells. So if this is a liver cell, then there's going to be glycogen molecules stored inside. So glycogen is made of glucose subunits. So let's label those subunits glucose. And the entire molecule, it's not going to write for me there, the entire molecule, if you want to indicate that, is glycogen. I'll move it up so I can write it. It's not going to write there. So the entire molecule is glycogen. Now, if it's been a while since you've eaten, then your cells are running low on glucose, and more glucose needs to be delivered to them, and it's delivered via the blood. So your body's going to respond by having your liver cells break up this glycogen into glucose subunits, and I'm going to add one here, and break it off. And then the glucose is going to be released from this cell into your system, where eventually the blood is going to deliver it to the cells that need this glucose. So that is what I mean by the result is going to be a release of glucose into the bloodstream. And this can happen because your blood sugar levels have fallen because it's been a while since you've eaten, or it could be due to stressful times or something scary, such as a giant bear chasing you. In this situation, the adrenal glands that are located at the top of the kidneys are pumping adrenaline into your system. And adrenaline molecules are going to cause your liver cells to respond in the manner that I just indicated. There's glycogen stored, and if you're scared, you're going to need a lot of glucose because you're going to need a lot of energy. So glucose is broken down into ATP. Your system's going to need more glucose. So in this situation, adrenaline is going to cause the breakup of the glycogen in the liver cells, and the glucose is going to be delivered to cells that are in need, like your muscle cells that are running and your brain cells that are trying to process and figure out how to survive. Now, let me note here, I indicated that if something scary, like a bear is chasing you, adrenaline is going to be released into the body. So in this situation, this messenger is adrenaline. But the more scientific term for adrenaline is called epinephrine. As a matter of fact, if you know someone who has an EpiPen, what is in that EpiPen is adrenaline. It's epinephrine, and that's why it's called an EpiPen. It's short for epinephrine. If your blood sugar levels are falling because you haven't eaten for a while, then what's going to be released is a hormone called glucagon. So let's put glucagon here. And glucagon is going to be the signaling molecule that's going to target the liver cells. Now, I'm going to go through all the steps of the signal transduction pathway that first starts with this G protein linked receptor, the reception, the transduction, and the response. And I'm going to reference a lot of big words. Remember, if you can reference these big words and use them in explanation, that's great, but you just need to know in general what happens whenever a ligand or a ligand, however you want to pronounce it, binds to these G protein linked receptors. So let's first start with the reception part. Now, here is that G protein that I keep referencing. This G protein is linked to this receptor. So here's the receptor, and initially, this G protein is here. It's linked to that receptor. Whenever the signaling molecule binds to the receptor protein, then this G protein becomes activated. And remember, it's the shape change of this receptor protein that is allowing for this G protein to become activated. Now, in order for this G protein to be fully activated, what happens is it has a molecule, it's called guanosine diphosphate, really similar to ADP. So it has guanosine diphosphate bound to it, and it trades that out for a GTP molecule, guanosine triphosphate. Those are details that are unimportant for you to be able to regurgitate back to me. Just note that whenever there's binding by the signaling molecule to the receptor, it activates the G protein that is linked to the receptor. That's all you really need to know. Now, once activated, this G protein moves and it activates another molecule that's in the membrane. In this situation, it's an enzyme, and this enzyme has a name. It's called adenylyl cyclase. Again, you don't have to have the name of this memorized. You could just simply call it, refer to it as the enzyme that's going to activate the secondary messenger. And that's what it's going to do. This enzyme binds to ATP and it strips off 2 phosphates. So I'm going to show 2 phosphates being removed. And what is left are these 2 phosphates and the secondary messenger that we've referenced before, cyclic AMP. Now, cyclic AMP is going to activate a molecule that's called protein kinase A. And how it does that is protein kinase A is regulated. That means it's turned on and turned off. And whenever nothing has bound to the receptor protein, protein kinase A is off. And it's off because it has these regulatory subunits. So I'm just going to label those as subunits. And there's more about them in the text on this page. But these are these regulatory subunits, so maybe we should put REG there for regulatory subunits. And cyclic AMP removes those. So whenever protein kinase A, so I'm going to put pKa, has those regulatory subunits removed, now it's active. And this kinase is going to go activate another kinase. So let's draw another kinase here. That's going to be activated by protein kinase A. I just put a big K there to represent this kinase. Now, I'm going to show you some other diagrams that show you what I'm about ready to talk about, but I want to go ahead and put it here so it's all on one page for you. What kinases do is they activate other kinases by phosphorylating them. That means that they put a phosphate on them. And when this kinase is phosphorylated, that means it had a phosphate group added to it, it is now active. So you now have an active kinase. And that phosphate group comes from ATP. So this kinase, pKa, that we first talked about, is going to bind to ATP and this inactive kinase, and it strips off one of the phosphates and attaches it to this second kinase, activating it. And down here, I'm attempting to show you how that occurs. So here is this kinase A, pKa, protein kinase A, that was activated when those regulatory subunits were removed. And so this pKa molecule is going to bind to another kinase. It's going to bind to ATP in the active site, and it's going to transfer a phosphate, and you see it right here. So this phosphate was on ATP, and now it's on this kinase molecule that's now activated. And as a result, ADP is generated. Eventually, a final type of kinase is going to be activated, and this activates an enzyme that breaks down the stored glycogen that is in this liver cell. And this also happens in the muscle cell as well, so that's why I have muscle. So this enzyme, let's kind of diagram that. So here's glycogen. So this activated enzyme is now going to break the bonds between those glucose subunits, and then that glucose can be released from this liver cell. Glucose is a polar molecule, so it's going to have to go through a transport protein. And there's different types of transporters that can transport glucose, and they're all called GLUT. They'll be like GLUT4 and GLUT1. But just in general, know that glucose is transported through these GLUT transporters. Now, I know that was a lot of information that we just went through, but I want to make sure you understand that it's all summarized for you right here. And again, some of the bigger words are not necessary to have memorized, just the general overview of this process. So again, here's a situation where we have a receptor that has a G protein linked to it. During reception, a ligand, a signaling molecule is going to bind to the receptor protein. It's going to activate this G protein, which is going to activate another enzyme. This enzyme is called adenylyl cyclase. Adenylyl cyclase is going to activate that secondary messenger, cyclic AMP. It takes ATP and removes 2 phosphates. Once cyclic AMP is activated, it can activate that first kinase, protein kinase A, which will go on to activate other kinases in the cell. Eventually, an enzyme is going to be activated, and this enzyme breaks up glycogen into glucose subunits that can be released through the GLUT transporters. Now, I want to show you two more models that show this process, because I can guarantee that you're not going to have a diagram on your quiz or your test that looks just like this diagram. There's so many different ways that you can model this signal transduction pathway. So take a second to run through this top model here and see what features it has in common with the model or the diagram that we just discussed. So again, we're going to have reception of the signal because we have a receptor protein. In this case, the signaling molecule is glucagon. And here's the G protein that's linked to the receptor. And whenever the glucagon binds to this receptor protein, there's a shape change. And that allows this G protein to activate adenylyl cyclase. So this isn't showing you that the G protein exchanges the GDP for a GTP. Again, that's a minor detail, and it's not something that you have to have specifically memorized. But here you see, once activated by the G protein, this enzyme that's embedded in the membrane can take ATP and convert it to cyclic AMP. And again, this is that secondary messenger. And from here, the signal is going to be spread from molecule to molecule. So it's going to be transducted. And as it's transducted, it's going to be amplified because one cyclic AMP can activate many protein kinases. Now, I wanted to show you this diagram because this diagram diagrammed those regulatory subunits that I mentioned on the previous diagram. And they are right here. So let's label those regulatory subunits. And this part right here is actually what is the protein kinase A molecule. So cyclic AMP just removes these regulatory subunits. And now this protein kinase A is activated. This molecule has the ability to bind to another type of kinase, like kinase B, and to ATP, and it removes the phosphate from ATP, adds it to the protein kinase B, which now activates it. So you need to remember that kinases activate other kinases, and eventually some target will be activated in the cell. And activating this target is going to lead to some kind of response, like I mentioned, activating a transcription factor, or activating an organelle, or a transport protein. But in this case, with glucagon and epinephrine, there's an enzyme that's getting activated, which can break up glycogen in those liver cells. Now, move down to the next model, and you're seeing the exact same process. Again, it's just modeled for you in a different way. One thing that you maybe have noticed is that this G protein looks different. There's actually 3 different subunits to the G protein. Again, minor details not to have memorized. And whenever there's the shape change by this receptor protein, it's the alpha subunit that actually activates the adenylyl cyclase embedded in the membrane. Now let's go down here to the end result to see if you understand what they're showing you here at the bottom. So I'm going to move this down a little bit. Remember, glycogen is made of many subunits, and that's what this N is indicating. So let's say glycogen is made of 1,000 glucose subunits. So that N is representing 1,000. And if this enzyme that is now activated, this is the enzyme that breaks up glycogen and glucose subunits. Once this enzyme is activated, it's going to remove those glucoses. And that's what this is trying to indicate. So this phosphorylase molecule is going to take one glucose subunit away from this glycogen. So it's N-1. And it doesn't just take one glucose subunit. It'll continue to break up, so you have many glucose subunits to release. Now, I like this diagram because it shows you that one enzyme can have many different jobs. So here, protein kinase A is activating this molecule, but it's inhibiting, or it's making this molecule right here inactive. So protein kinase A is inactivating this glycogen synthase A molecule. So that's why it's showing you that it is now inactive. Let's think about why we want this protein kinase A to inactivate this molecule, this glycogen synthase. So the name of this enzyme indicates what this enzyme does. This enzyme synthesizes glycogen. Well, We want glycogen to be broken down in glucose subunits and not glucose being assembled into these glycogen molecules. Because remember that the body has produced glucagon, or it's releasing epinephrine because it knows that your system is going to need an abundance of glucose for energy for whatever reason. Now, I just showed you 3 different models that show a signal transduction pathway. And these are all the same type of signal transduction pathway that was utilizing these G protein linked receptors. But there are countless signal transduction pathways that can take place in cells, and we don't have them all mapped out yet. But if I'm giving you a different type of signal transduction pathway, you need to be able to relate the ideas that you learned here with these examples to this other example to figure out what's going on. There's always going to be a reception, a transduction of the signal, and then a response by the cell. And remember, I mentioned that signal transduction is a big thing in AP. And here's the reason why. If you have a disruption in a signal transduction pathway, just one of them, then you probably have some kind of illness, some kind of disorder. A lot of medical research involves determining these signal transduction pathways, figuring out how they've been altered in people that are sick, and how we can fix this pathway. Even cancer is due to an alteration in a signal transduction pathway. And since you guys are brainy AP biology students, we want to teach you these ideas and these concepts now, because we hope some of you will go on to major in professions where you're trying to learn about signal transduction pathways and fix them so that we can get a cure for disease A, B, C that we do not have a cure for as of right now. No pressure or anything, right? You're going to find that in several units we're going to continue to talk about signal transduction pathways, and I'll continue to build on this idea and show you more concepts related to it. But I just wanted to give you a general introduction here while we were talking about these receptor proteins and how these receptor proteins can give a signal to the cell to respond. And here's the last page for today. If you remember, I indicated general functions of the cell membrane, and the last one I indicated that membrane proteins are important in anchoring or adhering cells. So here's a diagram that shows you that concept. We have 2 cells and they're anchored together. This is a membrane protein. Here's another membrane protein of the other cell. And you can see that these membrane proteins are adhering together, but they're also interacting with something inside the cell. They're interacting with the cytoskeleton. So the anchoring of these cells, or the joining of these cells, also involves the cytoskeleton inside the cell. Now I want to talk about the 3 types of animal junctions where there's membrane proteins that are binding these cells together, and then there's one type of plant junction to know. This diagram is out of your Cliff's Notes review book, and it's given you the 3 types of junctions that we find between animal cells. So we have adhering junctions, there's tight junctions, and there's gap junctions. Now, desmosomes are a type of adhering junction, and it's not important that you have them memorized, but you need to know more that when there's an adhering junction, cells are attached together and there's membrane proteins involved. So here's the membrane proteins. And adhering junctions keep cells together, but it also allows them to be flexible so that these cells and these tissues can stretch. So think of your skin cells. They have adhering junctions that bind one skin cell to its neighboring skin cells. These cells need to be bound together to form a barrier between you and the outside elements, but your skin also has to be flexible. So these adhering junctions allow some flexibility. So this tissue can stretch, but it's not going to break apart. Now, only certain specialized cells are going to have tight junctions. So imagine these are cells that are found lining your stomach. So here would be the stomach contents. So that's where your food's going to be after you eat. And we have our cells there, and those cells eventually are a barrier to your body cavity. Body cavity. And it's imperative that the acidic contents of your stomach aren't leaking out into your body cavity. So that's where these tight junctions come in. molecules, substances, cannot move in between those cells. And again, not all cells need to have these tight junctions to prohibit the movement of substances from one part of the body to the other. And then we have the third type, which are the gap junctions. And think of gap junctions as little hallways where molecules, cytoplasm, substances can pass from one cell to the next. So substances can move from one cell to the next through these gap junctions. And again, these gap junctions are due to certain types of proteins that are in the cell membrane of these cells. There can be chemical signals in one cell, so let's add some chemical signals, or just put chemicals in one cell, and it can activate other processes in the adjacent cell when those chemicals spread to the adjacent cell via those gap junctions. So I want to highlight this cell signaling and communication between cells. Because if it wasn't for these gap junctions, then chemicals and electrical signals wouldn't be able to move from one cell to the next. Now, for plants, I need you to remember that the gap junctions that we call in animal cells are called plasmodesmata. And plant cells. And this is a term to make sure you remember. I see it come up in questions quite often. So plasmodesmata in plant cells is like what gap junctions are to animal cells. They allow for the movement of substances between cells, and that's important in cell signaling and communication between cells. And then let's mention the extracellular matrix, just so that you know that the environment that's outside of a cell, so here's the cell's membrane and here's the cytoplasm. This is what I mean by the outside environment, or the extracellular matrix. And it's an aqueous solution that has polysaccharides and proteins. Molecules can move through it to the cell, from the cell. And you can see by the diagram that some of these proteins and polysaccharides can interact with membrane proteins. The interaction of these proteins might result in some kind of cell signaling event, or some of these interactions help anchor the cell.